Welcome to the lecture series in biochemistry for medical students. Good morning to all of you. Now we'll see something about the protein and amino acid metabolism. Now we know that proteins are polymers of amino acids, and all the body proteins, they are in a dynamic state in which new proteins are being synthesized, and after their lifespan they are being degraded and further synthesis takes place. So they are in a dynamic state. Now these amino acids that may be produced in this manner undergoes transamination and deamination reactions. Now the major two transaminase enzymes are GPT and GOT. Now GPT means glutamate pyruvate transaminase, also known as alanine transaminase or ALT, and GOT means glutamate oxaloacetate transaminase, also known as AST, that is aspartate transaminase. So these two transaminases are important in the various transamination reactions, and with these it is possible to synthesize non-essential amino acids. For example, in GPT you need glutamate plus pyruvic acid. Now the amino group of the glutamic acid is taken up by pyridoxal phosphate, the coenzyme, to become pyridoxamine phosphate, and this pyridoxamine phosphate then donates this amino group to the pyruvic acid, so that pyruvic acid becomes alanine and glutamic acid becomes alpha ketoglutarate. So in this manner, if we have excess of glutamic acid and there is a shortage of alanine, then alanine can be formed by taking the amino group from the glutamic acid to pyruvic acid, that means transferring the amino group. So that's why it is known as glutamate pyruvate transaminase. Pyridoxal phosphate acts as a coenzyme for this, and it forms first pyridoxamine phosphate. Now in the case of glutamate oxaloacetate transaminase or GOT, we need glutamic acid, then oxaloacetic acid, and in presence of this enzyme and pyridoxal phosphate again, the amino group from the glutamic acid is taken by the pyridoxal phosphate to form pyridoxamine phosphate, and then it is transferred to the oxaloacetic acid, so that aspartic acid and alpha ketogluteric acid can be produced. Now in this manner it is found that these reactions are reversible in nature. So ultimately it is found that all the amino group from the amino acids are rather concentrated in glutamic acid, and this glutamic acid further undergoes oxidative deamination with the help of glutamic acid decarboxylase or GAD, and glutamic acid dehydrogenase, not decarboxylase, glutamic acid dehydrogenase, and this forms alpha ketogluteric acid and ammonia. Now this ammonia is a toxic substance, and it has to be detoxified, and the detoxification of ammonia takes place in the liver. So liver has got a major role to play in the detoxification of ammonia. Now ammonia is highly toxic to the brain, because this ammonia can enter the brain and can consume again the alpha ketogluteric acid, so that glutamic acid can be produced, and glutamic acid further can be reacting with another molecule of ammonia to form glutamine. Now this leads to a less amount of alpha ketogluteric acid in the TCA cycle, and this may adversely affect the energy production. Hence, it is highly toxic to the brain. Now, this ammonia in its turn combines with, in presence of ATP and water and N-acetyl glutamate can be converted to carbamoyl phosphate. In presence of the enzyme carbamoyl phosphate synthase 1. So in this manner carbamoyl phosphate is produced. So in this manner the toxicity of ammonia is decreased to some extent. Now in the next step, the carbamoyl phosphate further reacts with ornithine in presence of the enzyme ornithine transcarbamylase, and it forms citrulline. Now citrulline further is acted upon by citrullinase enzyme, and then it forms arginosuccinate. Arginosuccinic acid is produced. Now arginosuccinic acid is further converted by arginosuccinase enzyme to arginine and fumarate. Now the fumarate can further go to the TCA cycle. Now the arginine is further acted upon by arginase enzyme, and what we get is urea, and ornithine is regenerated. Now this is how the ammonia is getting detoxified by various steps to urea. So urea is a harmless product, and urea is excreted in the urine, and all of us are ureotelic organisms, whereas there are uricotelic organisms like reptiles and birds, where the amino acid catabolic end product is uric acid, whereas in humans and other mammals it is the urea that is excreted. So birds we can understand because they have to fly, and so the weight should be decreased maximum, that means the weight should be least rather. Uric acid is least soluble in water, and so the weight of the birds can be reduced, whereas urea requires a lot of water because urea is soluble in water. So this is how the ammonia is detoxified. Now any defect in these reactions can ultimately lead to accumulation of ammonia, and these are known as urea cycle disorders. So there are different types of urea cycle disorders. First is when there is an absence of carbamoyl phosphate synthase, and some of the other enzymes can be decreased so that ammonia intoxication can take place. So this is about the catabolism of amino acids and the formation of urea. Now in the next topic I would like to tell you something about of course a different topic, that is the DNA recombinant technology. Now DNA recombinant technology is one of the most important molecular biology techniques carried out, and for this various tools are required. The first one is the vectors like plasmids, cosmids or bacteriophages are required. Then we need the molecular scissors, which are otherwise known as restriction endonucleases. Now these various tools are required. Now first we should have the target DNA, and the target DNA can be isolated of course, but then the isolation of the DNA is a very laborious process and very difficult process, So it is better we convert the messenger RNA into the DNA, into a single strand of DNA with the help of reverse transcriptase enzyme, and from the single strand of DNA we can get the double-stranded DNA molecule. Now this double-stranded DNA molecule, for example that of insulin gene, we have synthesized, and then insulin gene is to be further incorporated into a plasmid, which is a vector. Now both the vector and the insulin gene are processed with the restriction endonuclease, so that there will be base complementarity generated both in the insulin gene and also in the plasmid, so that re-annealing can take place and recombination can take place, so that the recombinant plasmid can be produced. Now this recombinant plasmid carrying the human insulin gene can be incubated with the bacteria Escherichia coli, and some of this recombinant plasmid and also the plasmid without any human DNA will be imbibed by the E. coli bacteria, and now we have to isolate the bacterial colony which actually carries the recombinant plasmid. Now this is not a very efficient process, only about 5% success rate is there. So we have to identify those particular bacterial colony which has got the insulin gene in the bacterial plasmids. Now plasmids actually confer antibiotic resistance to the microorganism. There may be antibiotic resistant genes for tetracycline, ampicillin, and also for chloramphenicol. Now if we grow wild bacterium in a medium containing ampicillin and tetracycline, then it can be seen that only those bacteria which has got a vector, which has got a plasmid, will be able to survive. Now, but there may be about 4 to 5 bacterial colonies in this manner. But now, if this is replica plated to another second plate, which also contains additional chloramphenicol, then you can see that one or two of the colonies which was in the original plate is not seen now in the replica plate, because they are dead. Now the reason for this is that the chloramphenicol has killed this bacterial colony. Now what is the reason for this? The reason for this is that the portion of the plasmid which is responsible for the chloramphenicol resistance is split by the incorporation of the insulin gene into it, and so that portion which confers antibiotic resistance of chloramphenicol is damaged, and so the bacteria all die. Now, so the correct colony which we have to isolate is the one that is the one in the original plate, that is corresponding to the dead colony. So in this manner, the selection of the bacterial colony can be done, and these bacteria can be subjected to produce more insulin protein, and then this can be used for the treatment purposes. So in this manner it can be done. Now yet another topic which I would like to discuss, which is also somewhat not coming in this group, that is the collagen disorders and collagen formation, their functions and their disorders. Now collagen is a structural protein. It's an insoluble protein. It is fiber-like, and to break a collagen fiber of 10 millimeter diameter, we have to 1 millimeter diameter. We have to apply a weight of almost 10 kilogram of weight. So it's a very strong molecule. Now the collagen is synthesized in the fibroblasts, not as mature collagen, but it is synthesized as the tropocollagen. Tropocollagen consists of 3 polypeptide chains, and each one of 1000 amino acids, and they are 3 polypeptide chains form a triple helix, triple quarter staggered structure, so as to make it very strong. But of course tropocollagen is not that strong enough. It has to be maturing into a strong insoluble collagen. Now this fibroblast, after formation, glycosylates some of the amino acids, and then it is secreted out, and then outside the cells certain peptidases remove some amino acids from the N terminus and the C terminus in a process of post-translational modifications. So some amino acids are removed, and this tropocollagen now becomes a procollagen molecule. Now in the next post-translational modifications, the lysine and proline are hydroxylated to hydroxylysine and hydroxyproline. Now again, one point which we have to remember is that every third amino acid in the collagen molecule is glycine, and there are quite many number of lysine and proline present, and some of these lysine and proline are hydroxylated to hydroxylysine and hydroxyproline. Now these hydroxylation reactions actually require the iron in the ferrous state, and to keep the iron in the ferrous state we need to have the vitamin C, which is a reducing agent. So in the deficiency of vitamin C, it is found that the collagen is not formed properly, and so scurvy can result due to the injury to the capillary walls. Now it is to be mentioned that collagen is the connective tissue protein in very many tissues like bones, cartilages, tendons, and walls of the blood vessels, and lens also. So the walls of the blood vessels are not properly formed, and hence there is a bleeding tendency. So collagen in this manner is a structural protein. Now there are various collagen disorders. One of them is osteogenesis imperfecta, and this is because one of the glycine molecules are replaced by cysteine, and hence the proper array of collagen fibers are not formed. And the cross-linking of the collagen is also affected, and copper deficiency also can lead to defective collagen formation. So osteogenesis imperfecta is a genetic condition where the bones become brittle and the joints are hypermobile, and so even a minor fall can cause fractures in such children. So this is one collagen disorder, and there are so many other collagen disorders. Another one is dermatoparesis, yet another one is Ehlers-Danlos syndrome, another one is lethaerism, and so many are present. And now lethaerism is due to eating of certain fava beans, which contains a toxin to some of the enzymes required for the maturation of collagen. Now homocysteine excess may also lead to collagen disorders. Now homocysteine, if it is in excess, can replace one of the lysine moieties, and then it can lead to an injury to the blood vessels, and where a thrombus can be developed, and so the homocysteine excess can cause myocardial infarction. So it's a risk factor. Homocysteine excess is a risk factor for the myocardial infarction. Now, collagen is also known as a permanent protein because there are no enzymes to degrade collagen, and the collagen formation starts from the beginning of the life itself, and then goes into the old age, and it causes excessive cross-linking, and in the old age there are this results in the problems of blood vessels and also in the skin. That is the old age problems. But in diabetes mellitus, sometimes there is a diabetic foot, and if there is an injury, and this if the bacteria like Clostridium histolyticum enters the wounds, and it may get septic and more infected, and then this is known as a gangrenous state can be produced in this manner. Because these bacteria, that is Clostridium histolyticum, they contain a collagenase enzyme which can destroy the collagen structure. So the collagen structure is known as a quarter staggered triple helix, and it is a structural protein which has to be, and this collagen also forms about 25 to 30% of all the total proteins in the body. Okay, thank you.